My name is Matteo Santin. I'm a professor of tissue regeneration at the University of Brighton. The Center for Regenerative Medicine and Devices, of which I am the director, has its roots in over 30 years of experience in research in the field of biomaterials. We have developed biomaterials for orthopedic implants, cardiovascular devices, for ocular and middle ear implants, as well as for extracorporeal devices, including bioartificial liver. Also, our knowledge about the interaction of the cells with the materials has enabled us to move further in our research, whereby rather than trying to replace damaged tissue with the traditional medical implants, we could push our ambition trying to regenerate rather than replace the actual tissue, and to do that with the approach which is known as regenerative medicine. At the same time, this knowledge between cell and material interactions has led us to produce organoids, or if you like, organ on chip, that enable pharmaceutical companies as well as fellow researchers to study the cells more into details and to test novel drugs, reducing the animal experimentation.
My name is Giordana Peregrino and I'm a PhD student at Brighton University. The aim of the project is to design and develop a new carrier for the delivery of a chemotherapeutic drug and nucleic acid for the treatment of an aggressive breast cancer subtype. Unfortunately, breast cancer is one of the most common women cancer, and the subtype that I focus on, that is called HER2, has a lower survival rate than other subtype. The design nanovector has a branch structure that is able to bind and protect the drugs from the immune system and the extracellular environment. Nonetheless, it is equipped with a targeting sequence which recognizes the cancerous cells and facilitates the uptake. The carrier for both the traditional chemotherapeutic drug and the nucleic acid has an nanodimension that is similar to a protein, and it is necessary to increase the stability of the cargos and to concentrate the drugs into cancerous cells, thus reducing the adverse side effects to patients, and won't activate the immune system, which can damage the drugs and remove them from the blood circulation.
Hello, I am Dr. Paul Harris, and I am a reader in applied mathematics here at the University of Brighton. One of my current research projects is in the development of mathematical and computational models of chemotaxis. This is where cells move in response to changes in the concentrations of chemical signals in the surrounding medium. In the first models that I developed, the cells were considered to be rigid particles and the effects of the chemical signal modelled as forces acting on the cells. The motion of the cells is simulated using the classical equations of motion originally developed by Newton, amongst others. The video playing on my right shows the results of using a mathematical model to simulate how individual cells and small clusters of cells come together to form larger clusters. However, whilst this model is good at simulating how cells come together to form clusters, it does not give any information about how individual cells move. They do this by deforming their outer membranes in the direction in which the chemical signal is increasing. The body of the cell will then follow the deformed membrane as the cell returns to its original undeformed shape. In addition, the research is investigating how being part of a cluster affects the way in which the cell reacts to the chemical signal.
Damage to intervertebral discs is a leading contributor to disability worldwide. Current disc replacements in the form of artificial devices are typically made from polymers and metals. However, many of these artificial discs are unable to comply with the mechanical motions of the spine and can damage adjacent tissue. Our work focuses on the regeneration of intervertebral discs using bioprinted scaffolds to mimic the mechanical and physiological behavior of the human spine. We're also using this approach to show how disc degeneration affects the behavior of the spine. This will help surgeons and health professionals see and feel these differences with real-world models to hone surgical techniques and improve healthcare.
My name is Peter Watt. I'm part of the Center for Regenerative Medicine and Devices, I'm a physiologist and biochemist at the University of Brighton, and I have research interests in metabolism, health, and things like diabetes and obesity. Exercise is easily recognised as a good thing to do for your health and to reduce risks of all sorts of diseases and ailments. It's clear that similar benefits arise because of adaptations to doing exercise, such as your increased heart rate, metabolism and blood flow around organs of the body during the exercise. And our work is aimed at understanding some of the new insights that have come out of regenerative medicine research. We're examining in particular the effects of exercise on stem cells, stem cell function. These are cells that are involved in tissue repair and growth and are affected directly by exercise, such that after exercise you can see an increase in the number of these cells in tissues and in your circulation. One example of our work aimed at this particular issue involves Achilles tendinopathy. Achilles tendon is the one in your heel, and it can become swollen and can even rupture. When it ruptures, you have to undergo a long period of therapy to get it fixed. What we're trying to do is to look at how exercise can stimulate the regeneration of the Achilles tendon after such a problem. Using sensitive ultrasound equipment we've acquired, we can measure the strength of the Achilles tendon and examine how it responds to different types of exercise and how quickly it can repair. We have shown that placing a load on the injured tendon can speed repair and recovery to normal function quickly and effectively. What we aim to do next is to use our understanding of stem cell biology to see how we can enhance this process and make recovery even quicker and more effective.
Here at the Clinical Imaging Sciences Centre, we use our state-of-the-art MRI scanner to answer some of the research questions important for the Centre for Regenerative Medicine and Devices. Our research team have created methods that transform the scanner from being an impressive camera into an incredible measurement tool that provides sensitive biomarkers capable of monitoring damage, disease, and recovery anywhere in the body. As members of the CRMD, we've provided vital contributions across a range of disciplines to objectively quantify fatigue in multiple sclerosis, tissue degeneration in motor neuron disease, as well as the development of debilitating cysts in the spinal cord. Staying with the spine, we've also used our scanner to measure the water content of healthy and damaged intervertebral discs. We also used spectroscopic methods to probe the chemical composition of these discs. These metrics are likely to be crucial to the successful generation of disc implants in future CRMD projects. Another area of focus is the blood-brain barrier. This is a semi-permeable interface that protects the brain from neurotoxins that may be circulating in the blood. We've developed a technique that can determine the permeability of the blood-brain barrier in order to study the impact neurodegenerative diseases have on this vital defense. Another exciting avenue for our research in the CRMD is with metabolic imaging, where we can use our scanner to detect phosphorus concentration to study ATP utilization. This will open a window into cellular energy metabolism. Together, we provide a formidable range of in vivo and ex vivo quantitative imaging and spectroscopic methods to tackle many challenges in the field of regenerative medicine.
Hello, my name's Rachel Force. I'm a senior lecturer at the University of Brighton in the podiatry department, and I'm currently working towards a PhD by publication. My PhD is focusing on wound exudate viscosity and how this affects the performance of commercially available wound dressings. As a member of the Centre for Regenerative Medicine and Devices, I'm a key member of the EPSRC study. The study is looking at 3 different types of dressings, and we are going to look at what happens at the contact surface between the wound bed and the dressing surface. We will be recruiting patients with new wounds and following their healing journey, some from start to finish, some a snapshot in the centre. And the tests will all be performed on the dressings once they have been removed from the patient. We will be looking for specific proteins and macromolecules that will be present, as well as inflammatory mediators. The information gathered will assist with future functionalisation of wound dressings and enable us to monitor wound inflammation and the healing process occurring in wounds.
In 2013, the University of Brighton recognized the quality of our research and granted us with the status of a Center of Excellence. We are really committed to the global challenge agenda set by our university. In fact, our research focuses on clinical conditions that affect the population worldwide. We hope that in the years to come, we could really contribute to the scientific community as well as to our wider population with our innovation, knowledge, and commitment to all.